Hello everyone, welcome to a session on interaction of genes. So this is the second module that we are going to discuss in genetics. In this particular session, we are going to see how genes interact with each other. So in Mendelian genetics or Mendelian principles, it was stated that two alleles interact together to produce a character. And after the rediscovery of Mendelism or Mendelian principles in the early 1900s, many works or many scientists who worked on this particular field of genetics discovered that every alleles or every factor or every gene as discovered by or as described by Mendel, every factor does not or the pair of factor which interacts together to produce or produce a trait which was detailed by or described by Mendel. After the rediscovery, many scientists discovered that this particular law is not universal. There are interactions which are happening other than allelic interactions. Mendel, in his principles and in his laws, he discussed only about non or allelic interactions where the two alleles of a particular gene they interact together to produce a particular trait. But on further investigation after the rediscovery, it was found that there are many other interactions which are happening in the genes which leads to the production or the expression of a particular character. And this led to the proposal which is called as factor hypothesis or interaction of genes, which was proposed by William Bateson and Punnett. And they stated that the character need not be necessarily due to the action of a single factor or a pair of alleles, but may be also due to the action of several factors. And this is what they called it as factor hypothesis. That is, Mendelian law which described only the interaction between two alleles, which is leading to the expression or leading to the production of a particular character, cannot be universally applied to every genes. There are interactions other than Mendelian interaction. And the suggestion does not mean that Mendelian laws or principles are false. There are alleles or there are genes which follow Mendelian inheritance, but apart from that, there are also other interactions which results in the formation of a particular character or a particular trait. And they discovered mainly different or the different interactions come under mainly four heads. That is one character and one gene, where a character is controlled by a particular gene and its pair of alleles, that is the two alleles of a particular gene, but they do not depend on Mendelian dominance, or their expression is different from normal Mendelian dominance. They act differently in the dominance which was suggested by Mendel. That is the first one. Coming to the second one, which is two genes and one character. So in this particular interaction, it is a non-allelic interaction. First one was an allelic interaction, the interaction between two alleles as stated by Mendel, but they behave differently than Mendel suggested the Mendelian dominance. In the second one is the interaction between non-allelic genes, which was not described or proposed by Mendel. So this is a new one, that is the interaction between two non-allelic genes, that means two different genes. They interact together, but that interaction will result in the production of a particular character. That means a single character for say the color of a flower. So as suggested by Mendel, it was controlled by two alleles, but here they say that in some other plants or in some other organisms, it was proved that the production of a particular color, the production of a particular pigment, etc., is controlled by non-allelic genes. That means non-allelic genes, they interact together. That means there will be four pairs. That means there are two genes in such cases. They are non-allelic, and each gene will have two alleles each. That means two pairs of alleles, they interact together to produce a particular character. Such an interaction is called as, or such interactions are referred to as non-allelic interactions. And the third one is three or more genes and one character. It is more or less same as the second one here, apart from, or rather than two genes or two non-allelic genes interacting together. Here three or more non-allelic genes are interacting together to produce or to express a particular character. And the last one is multiple effects. Multiple effects is the reverse of what we have discussed in the above three. Here a single gene might be controlling or might be influencing more than one character. So these are the different, or these are the deviations from the normal Mendelian principles, and these are the interactions that are going to be discussed under this particular module, which is called as interaction of genes. I point upon one more thing, or stress upon one more thing, is that Mendelian laws is not proved to be incorrect. Or this particular chapter is not a proof for or proof that Mendelian laws are false. It is just an addition to Mendelian laws that apart from what Mendel suggested, Mendelian laws still exist. Mendelian principles, Mendelian dominance, etc., are still being inherited, or it is the pattern of inheritance in many genes, even in humans. But apart from that, there are other interactions where different genes interact together to produce a trait, and alleles itself, they interact in a different pattern, influencing a particular a dominance which is, or it does not follow the dominance which is suggested by Mendel. So this is the topic of discussion of this particular module, that is interaction of genes or gene interaction. So for the ease of our discussion, we are going to classify gene interactions into two: allelic interactions and non-allelic interactions. So in allelic interaction, as we discussed earlier, in allelic interaction, what we are going to say is that what is the interaction between alleles that they do other than Mendelian dominance. So Mendelian dominance is nothing but in a heterozygote, what happens is the color or the trait of one of the parent will be only expressed. That means if you have a red flower and a white flower, in the F1 heterozygote you have the red color, and hence you get the red color in the F1 generation. You call that red as dominant, and the white, which is not expressed, as recessive. But this is, or this particular red to red inheritance, that is from a red parent to a red offspring, that is not universal. There are other interactions happening between alleles, between these two, the alleles of a particular gene, to produce a character, and rather than the normal dominance which is suggested by Mendel. So first we are going to see the allelic interaction, and we'll in the next coming sessions we'll be seeing what are the non-allelic interactions. Coming to the allelic interactions, the allelic interactions are interactions between the alleles of a single gene, and here we are mainly going to discuss about the variations of Mendelian dominance. What are the different interactions that genes involve apart from Mendelian, or apart from the dominance or the dominant trait that is suggested by Mendel. So the dominance is a type of interactions between the alleles of a single gene. And here we are going to discuss four different types of dominance. The first one is complete dominance. So complete dominance, and the second one is the incomplete dominance. The third is codominance, and the fourth one is the lethal gene. So these are the four different allelic interactions we are going to discuss, and these four are variations of the normal dominance or dominance interaction that was described by or proposed by Mendel. So we'll be discussing one by one. The first one is complete dominance. So in complete dominance, we are going to discuss three different terminologies which we didn't come across in Mendelian inheritance, that is haplosufficient, haploinsufficient, and dominant negative type of interactions. So coming to complete dominance is the same as that described or proposed by Mendel. So we'll first see first we will recheck on what is complete dominance which was proposed by Mendel, and we'll move on to the variations. So complete dominance is a trait which is expressed in both the homozygous as well as the heterozygous condition. So if a particular trait is being expressed in a similar manner in a homozygous and a heterozygous condition, that particular trait will be called as a dominant trait. As you can see in this particular diagram, which we have already discussed, which is this one, which you see here. That is the interaction where, this is the red color flower and white color flower. When they are crossed, you get color flowers in the F1 generation. That means the red one is directly inherited to the F1 generation, and this red which is present in the F1 only is considered to be what is called as a is said to be the dominant trait, and the white which is not seen in the F1 is considered to be recessive. So in such cases, what happens is the homozygous dominant and heterozygotes cannot be distinguished at the phenotypic level. As you see here, you cannot distinguish this red to be different from this red. Both are the same according to the phenotype. Only the genotype is different. If you see the genotype, that's why we have not marked the genotype here. So if you are going to see the genotype, there is a difference. This will be homozygous dominant with two capital letters. If it is R, two capital R's, and this will be different. It will be having a capital R and a small r, but phenotypically both will be the same. They will be having the same phenotype, which is red. So this Mendelian dominance is shown by many this type of Mendelian dominance is shown in the case of many human disorders. One is a metabolic disorder, which we are going to discuss. It is called as phenylketonuria. So this particular disease or this particular condition is caused when you have homozygous recessive genes for this particular enzyme. So this is a metabolic disorder. When you get the recessive alleles in a homozygous condition, a particular enzyme for the metabolism of phenylalanine will not be produced, and that will result in this particular disorder, which is called as phenylketonuria. And this particular gene for this particular enzyme. It is because of, or this phenylketonuria is because of the lack of an enzyme in the metabolic pathway, and the lack of the enzyme is due to, or this particular enzyme can be produced only by the dominant allele, and the recessive allele does not have the capacity to produce that particular enzyme. But so what happens is, when you have a homozygous recessive homozygous recessive alleles, you will have this particular condition called as a metabolic disorder, which is called as phenylketonuria. But when you have the dominant gene in the homozygous as well as the heterozygous condition, you will not inherit that particular disorder. You will be normal. So in that case also, you cannot distinguish between an individual who is having a homozygous dominant alleles for phenylketonuria and the other one, the heterozygote, because both will be normal. So in this particular case, this is what is called as complete dominance, so which was described by Mendel. So this is one instance where a gene is following Mendelian inheritance, and in this particular case, we can see that to produce, so if you take this red color, to produce this red color, only half of the allele is required. That means in this particular case, this is a heterozygote with a dominant allele and a recessive allele. In this case, only half of the allele is required to produce this particular color, which means the red color. That means only a single capital R is having the influence to produce the red color. In such case, you can say that only half of that allele is required for the expression of that particular character or that trait. And such type of alleles or such type of condition is referred to as haplosufficient. Haplo means half. That means half of the allele is, or half dose of the allele is sufficient for the expression of that trait. That is called as haplosufficient. So haplosufficient is same as Mendelian dominance, where the homozygous and heterozygous will be having the same phenotype, and because the heterozygote is having the capacity to produce the trait at the same level as a homozygous dominant, you call it as haplosufficient, means half is sufficient for the expression of that particular trait. So this is This is just an explanation of what is our Mendelian dominance. But there are some other instances where the dominant allele do not show this dominance in the heterozygous condition. So this is what is called as deviation to the normal Mendelism or the Mendelian principles. So in such cases, what happens is, this is a normal one where you have the red color. The heterozygote also expresses the red color. But in other instances, you can see that the dominant allele or the dominant one produces a particular trait, but the heterozygotes does not have the particular trait, or it does not express that particular trait. And in the dominant trait is not expressed by the heterozygote. And usually it is seen that in such type of conditions where you do not have the homozygous dominant individual which show the particular trait, this particular situation is called as haploinsufficiency, which is just opposite to haplosufficient. So haplosufficient was the heterozygote as having the capacity to produce a particular trait or particular character of the dominant or homozygous dominant. But in this particular case, which we are going to discuss in detail, this particular case, what happens is that the dominant or the homozygous dominant individual only show that particular character. When coming to the heterozygote, it does not show that particular character, it shows another character. And in such cases, you can say that that particular allele is haploinsufficient. That means it can have it has the capacity to express only in the homozygous dominant condition. So in haploinsufficiency, one dominant allele is not enough to achieve the normal function. So if you take an example of haploinsufficiency, it can be stated in a more or less a simple elaboration like this. So take a case or take a hypothetical situation where you need 18 protein which is required for a particular reaction, for say a particular biosynthetic pathway or a metabolic pathway. And for that pathway to be normally function or to function normally, it requires 18 molecules of that enzyme. And consider a dominant allele is having the capacity to produce, or it can produce only 10 molecules for a particular reaction, or it is capable of producing 10 molecules. So if you have two dominant alleles, which is the homozygous dominant condition, if I am homozygous dominant for that particular allele, what happens is there will be, or my cells will produce 20 molecules. So 20 molecule is more than minimum. 18 is required for the normal function and 20 is more than minimum. As a result, homozygous dominant will not show that particular, or will not show any deficiency of that enzyme. They will be normal. But just imagine what happens when you are heterozygous. So when you are heterozygous, what happens is your dominant allele will produce. So heterozygous means you have one dominant allele and the other one is a mutant or a recessive one. So that will not produce any particular protein, or it produces a protein which is non-functional. So here in this case, what happens is if you are a heterozygote, the dominant allele produces the so-called 10 units, but the heterozygote produces, in this particular case, the heterozygote produces a protein. But that particular protein is a non-functional protein. Okay, it does not have, it does not function as an enzyme. Even though it produces a complete protein, it is a functionless protein, and that can be considered to be zero, because you have zero functional molecule from a recessive or a mutant allele. So in a heterozygote, you have 10 plus 0, which is equal to 10, and there the half is insufficient. You require a homozygous dominant condition to have the expression, or to have a normal function. And in such conditions are referred to as haploinsufficient. And such cases are usually due to a dominant mutation, and that produces a non-functional protein. So this particular condition, which is called as haploinsufficiency, is usually seen to be due to a dominant mutation. So these mutations are usually called as dominant mutations because If you observe, you can see that here the dominant or the wild type, usually the dominant, what you call it as dominant, is a wild type allele. Allele is said to be wild type. We have discussed wild type allele in the previous sessions. So a wild type allele is nothing but the allele which is seen more in a population. That means a normal, the people which are normal. If you take a disorder, the people which are having normal metabolic pathway are considered to be the wild type or the dominant one, and the disordered state or the diseased conditions are usually referred to as recessives, because they will be seen in less number when you consider a human population. So in such instances, this particular so wild type is usually called as dominant one. In every gene you call or you term it like that. What is normal is called as wild type, and that will be considered as dominant because that is the one which is expressed by more number of individuals in a population when you come to humans. So in this particular case, such mutations or such recessive characters which produces a non-functional protein. Here a protein is being produced. Here the particular mutant or the recessive one, the so-called recessive one, it is not restricted from producing a protein. It is producing a protein, but the protein is non-functional. It is a non-functional protein. And now this particular mutations, rather than calling them as recessive mutation, you call them dominant, because if you observe, you can see that the heterozygous, it has only 10 units, so it will produce, if it is a disease or metabolic pathway, that metabolic pathway will not function as, and hence it results in a disorder. And the same thing will be observed in a homozygous recessive condition. That means a mutant homozygous condition. The mutant homozygous means it also will lack the particular, it will completely lack the particular molecules, and hence it does not produce even 10 homozygous recessive. If you take the example, it does not even produces the 10 molecules. As a result, what happens is the recessive, that is homozygous recessive, and the heterozygous, it is just the reverse of the normal dominance. Here the homozygous recessive or the mutant, you don't call it as homozygous recessive, the homozygous mutant one and the heterozygote means a wild type plus the wild type and the mutant, they are producing the same phenotype, which is the disorder, because the protein, in one case in the homozygous mutant one, the protein is not produced. In the heterozygous condition, half of the protein is produced, but it is not sufficient for the process, and hence this is a type of mutation. Because when you have something in a homozygous condition and the same is being expressed in the heterozygous condition, you should call them as a dominant one, and hence this is called as a dominant mutation, or such inheritances are usually called to be a dominant mutant inheritance. So it is a mutant one, it is not wild type. If it was wild type, you just call it as dominant inheritance, but here dominance is reversed because of this mutation and haploinsufficiency, and hence such mutations are usually referred to as dominant mutation. So it is just the opposite. So opposite in the sense, you should remember that the homozygous mutant and the heterozygote will be having the same phenotype. That means they will have the rather than the normal dominant, the dominant inheritance that was suggested by Mental. So haploinsufficiency is responsible for one of this syndrome, which is called as Digeorge syndrome. This is a genetic disorder, mental illness. So this accounts for many symptoms in humans. So this is an example of this particular haploinsufficiency, which is Digeorge syndrome. So coming to the next one, which is the last one, which is dominant negative. So dominant negative is also due to a dominant mutation. So in this case, so this happens in special cases where a gene produces protein monomers, and for that protein to function, these protein monomers, they combine to produce homodimers. So dimer means a protein with a complex protein with 2 units which are of the same type. They are called as, if a dimer is formed of the same units, they are called as homodimers. So in some instances, a gene produces protein monomers which combine to form homodimers, and this homodimers are the functional one. They might be functional enzymes, they might be functional as structural components, etc. So only dimers will be functional. Monomeric units are not functional. So a gene produces protein monomers, and these monomers combine together after the protein is formed. They combine together to form dimers, and these dimers will be responsible, or they will be functional one. So in a mutant heterozygote, as you see here, so this is a normal one, normal monomeric units, they combine to produce a functional one. In a heterozygote, you can see such a condition. In a heterozygote, here also the protein is being produced, but the protein is a crippled protein or an inactive protein. So in the former case, we saw that the inactive protein does not function. It is compared to 0. But here what happens is, this inactive protein combines to the other monomer, that is the functional monomer, and renders this inactive. So these are the three conditions. This is a homozygous dominant, the normal one, the normal individuals, heterozygous one, and the homozygous mutant one. So here what happens is, the wild type, I don't call them as dominant and recessive. So this is the wild type allele. This is a heterozygous between the wild and the mutant one. This is the homozygous for the mutant one. So what happens here is, in the normal one, the monomers combine together, they form a dimer, and that dimer is the dimer functions normally. But here, rather than the non-functional protein being functionless, or rather than it not being attached to the functional protein, what this particular monomeric unit does is, it comes and binds to this monomeric unit and renders this inactive. So when all the monomeric units, which are the functional monomeric units, because half will be producing functional protein and the other half will be producing this non-functional or crippled protein, as a result, it produces the same effect as a homozygous mutant, because whatever functional protein is being produced, that will be inactivated by this particular one. As a result, this heterozygous and this homozygous mutant will have the same phenotype. That means they will be having that particular deficiency or that particular trait. And that is why it is said that the mutations which are haploinsufficiency and dominant negative are usually considered dominant to the wild type, because homozygous mutants and heterozygote expresses the mutant phenotype. That is what we see here. In this particular case, this one is interacting with this and rendering this inactive. But in haploinsufficiency, what happens is, the inactive protein is inactive, but the number of active protein is not, or the concentration of the active protein is not enough for that particular character to be expressed. So these are the three different types. The haplo-sufficient is the normal one to the normal one, or the normal Mendelian inheritance, and these two are the deviations from the normal Mendelian inheritance, and because of, or just the reversal of normal Mendelian inheritance due to dominant mutations. So dominant mutations, here you have to keep in mind that it is better not to use dominant and recessive alleles. You can call this one as wild type and this one as the mutant. So that will make sense. So rather than calling this as dominant and this as recessive, that makes confusion. So in our discussions in this particular Mendelian, in gene interactions, we'll go on with the terms like wild as well as the mutant phenotypes. That will make more sense than calling them dominant and recessive. So I hope this particular session is clear. So this was the first type of gene interaction coming under allelic interaction. So here these are alleles which are interacting. These are the genes or the different forms of a particular gene. One is the wild type and the other one is mutant. That's the only difference that you see here, but they are interacting in a different pattern than normal Mendelian inheritance or Mendelian dominance. So the others are the other ones which are the other allelic interaction, which are incomplete dominance, codominance, and the lethal genes will be discussed in the coming sessions. Thank you for hearing.